Good morning, everyone. Good morning, and welcome back to the part 3 lectures of one of the most important chapter, Principles of Inheritance and Variation. And today's class, we are discussing with the topic of Monohybrid Cross and Laws of Inheritance. Yes, you may have a echo right now. I'll clarify that. So if you have any just problem with the sound or anything, so please don't forget to inform me about the same. Okay, everyone. So in today's class, we will be discussing on the part 3 lecture of this chapter, Monohybrid Cross, Laws of Segregation and the Law of Inheritance proposed by Mendel. So without wasting our time, we are moving on to the chapter. Is that clear? Is that clear? Guys, can you all hear? Guys, can you all hear me? No rest. Yes, perfecto. So let's move on to the session without wasting our time. Yes, everyone. I hope you all can hear me. So, those who are watching the session right now, don't forget to hit the like button, do share it with your friends, and subscribe this amazing platform for Neet Biology. This is our amazing Telegram group t.me/neet_biopoint where daily session updates, regular discussion, daily polls, and interact with us takes place. We will have daily quizzes in our group sharp at 4:15 p.m. and 7:15 p.m. in the group. And those who are watching the session right now, once again reminding you, don't forget to hit the like button, to share it with your friends, and subscribe the amazing platform. And those who are watching this session, don't forget also to leave a comment in the comment section after watching this video on how do you feel this chapter or this topic after listening to the class. So let's get started. Before we get started, let's remind what we have learned in the previous classes, because long back we have studied everything and I know that most of you have forgot almost everything, right? So before we start, let me tell you a very few things which is needed throughout the chapter. You have to remember Gregor Johann Mendel is known as the father of genetics. Gregor Johann Mendel is known as the father of genetics. He used the term factors. He used the term factors for genes. Who coined the term genes? The term genes was coined by Johansson. Okay, the term genes was coined by Johansson. So Gregor Johann Mendel used the term factors instead of genes, because at this point of time when Gregor Johann Mendel was doing the experiment, there was no idea about chromosomes, nucleus, or any structures or even DNA. Yet he discovered heredity and variations which are seen in animals. So Mendel conducted some hybridization. Yes, 1 second. Yes. Mendel conducted some hybridization experiments. Mendel conducted some hybridization experiment on garden pea, that is to be Pisum sativum. Okay, Pisum sativum. This is a previous year Neet MCQ. For 7 years, previous year MCQ question. Okay, so Mendel conducted some hybridization experiment on garden pea or Pisum sativum for 7 years, that is from 1856 to 1863. This was a previous year MCQ and this was also a previous year MCQ question. So guys, this, if you are taking this you can take as to be the white flower, this is to be the violet flower. If In the question it is given like violet. We know that violet is the dominant, so you'll take capital V, and white you'll take that small v. Or if white is dominant, then capital W, small w. So the capital letter indicates the dominant trait and the small letters indicates the recessive trait. Is that clear? Yes. Next is the Mendel's laws of inheritance. Moving on to the Mendel's laws of inheritance, before we have to understand what are the steps which are involved in making a cross in a garden pea. You know that if we are taking a white colored flower, if we are taking a violet colored flower as the male parent, sorry, in this case, I'll tell in this case, perspective of this particular diagram given over here. So if you are taking this violet flower as the female flower and white flower as the male, what happens? This pea flower, that is from the violet flower, you have to emasculate. What you have to do? You have to first of all select. First of all, select the two pea plants with contrasting character. That is, in this case, we have selected two different character. That is, the flowers having violet flower and white flower. So we have taken the white flower as to be the female parent or the mother, and the white flower as the male. And after identifying the parents, we have to make sure that we done emasculation. What is emasculation? Removal of anthers from one plant to avoid self-pollination. So emasculation is done in female parent. Okay, emasculation is done in female parent. Emasculation is done in female parent to avoid the self-pollination. Okay, that is what is said to be the emasculation. One second. One second. One second. Okay, so you have to do emasculation. After emasculation, both the parents have to be pollinated. Okay, collection of pollen grain from the male parent and transferring it into the female. Okay, collection of pollen grains from the transferring it into the female parent is referred to as the pollination. The last step is the collection and germination of seeds to produce the offspring. Okay, I'll show you a cross. I will show you a cross. Here we are taking violet flowers as well as white flowers. Okay, so let me take out a blank slide first of all. Okay, we are taking the parent as violet flowers. We are taking a violet flower is crossed with white flowers. I am telling the cross first of all itself because I know that you all know about how to do a Mendel's cross. Okay, so violet flower we are taking it with capital V, and white we are taking it as small v. So in the F1 generation, you will get a progeny with capital V, small v, which is actually a violet progeny, which is actually violet in color. Okay, which is actually violet in color. Yes, and if you take this violet, if you self pollinate, okay, sorry, if you self pollinate this violet flowers, then you have to draw a Punnett square. Okay, capital V, small v, capital V, small v, capital V, capital V, capital V, small v, capital V, small v, small v, small v. So you will get 3 violet flowers is to 1 white flower. 3 violet flower is to 1 white flower. That is 75% of the progeny formed resembles one of the parents and only 25 represents the recessive trait. Whereas in F1 progeny, if in the question they are asking to you about the F1 progeny, then the F1 progeny is 100% to the dominant parent or one of the parent only. Is that clear? That's all about the Mendel's laws of inheritance. This is a table which we have been going on discussion from the first lecture onwards because this is that much of importance for your exam. That's why I keep on telling you to study this, study this, study this again and again because this is very, very important. First of all, let's take a stem height. That is tall is dominant, dwarf is recessive. Flower position axial dominant, terminal recessive. Flower color, yes, what we have done. Violet is dominant, white is recessive. Port shape, inflated, constricted. Port color, green and yellow. Seed shape, round and wrinkled. Okay, guys, round is capital R, capital R. Wrinkled is small r, small r. Okay, and seed color is yellow and green. Look here, tall and dwarf. So tall is indicated by capital T, capital T. Dwarf is indicated by small t, small t. Axial, capital A, capital A. Terminal, small a, small a. Violet, dominant, capital V, capital V. White, recessive, small v, small v. Constricted and inflated, inflated, capital I, small I. Or you can write capital C, small c. Capital G, small g. Here capital R, small r, capital Y, small y. So this is how you represent each and every alleles or the characters which are selected by Mendel as a true 7 breeding varieties. Okay. So we are moving on or entering into what we the topic for today. That is what we have to discuss for today. We are moving on to the topic that is monohybrid cross. In all the slides, actually, I know you know the concept about monohybrid cross, but you feel difficulty in doing the problem. So I've included a small 2 to 3 questions after the session. So at the same time, we would be having a separate numerical solving question or mathematical solving questions of this chapter at the last or as soon as possible. I will do that. Okay. So I know that most of you know the concept about the monohybrid cross and do I know very well how to do the cross of monohybrid cross, but you feel difficult in doing the concepts of test cross, incomplete dominance, complete dominance, dihybrid cross, especially pedigree analysis. All those things are the most difficult portions of this chapter faced by certain students. So monohybrid cross, the cross is favorite for everybody because it is one very easiest cross that you can come across in this chapter. So first of all, let's discuss about what is a monohybrid cross, and after that, let's do into some examples. Clear, everyone? So first is the monohybrid cross, a cross involving two plants differing in one character pair. I hope the annotation is clear. Okay, a cross involving two plants differing in one character. For example, if you are taking tallness, if you are taking tallness as the character, what happens? Capital T indicates tallness, small t indicate dwarf. Or small t indicate tallness, capital T indicates tallness, small t indicates dwarf plant. Exactly like that. If you are taking only one particular character for crossing, then that is said to be monohybrid cross. Mendel crossed tall and dwarf pea plants to study the inheritance of one gene. So in case of your question, they can ask you this particular sentence also. Inheritance of one gene. Okay, inheritance of one gene. Inheritance of one gene is similar to what monohybrid cross. Similar to what monohybrid cross. For example, tall versus dwarf. That is cross between a tall plant and a dwarf plant is an example for what? Monohybrid cross. Otherwise, you can do yellow versus green. Yellow versus green is also a monohybrid cross example. It's an example for a monohybrid cross. Okay. First of all, let's do a monohybrid cross and make it clear. Okay. In the first generation, we are taking a parent. Okay. First of all, for doing a cross, we have to select a parent. So for the parent, I am taking tall plant, homozygous tall, and homozygous dwarf. I am taking. Okay, I'm taking homozygous tall and homozygous dwarf. That is capital T crossed with small t. So second step while you are doing a cross is to write the gamete. For Neet preparing, you're completely focusing on Neet. You don't have to do all these things. You have to have a mind calculation for all these things. So capital T is the gamete over here, small t is the gamete. So these two are crossed and you will get the F1 generation. Capital T, small t, which is heterozygous tall. Heterozygous tall. And then what you are going to do is you are going to self-pollinate. Selfing of F1 generation. That is capital T small t crossed with the capital T small t. That is explained with the help of Punnett square, which was proposed by Reginald C. Punnett. Okay. Capital T small t, capital T small t. What is the other term for this? Punnett square. Okay, this is what is said to be what? Punnett square. Or otherwise you will call them as checkerboard. Okay, how are you doing? Going to do the cross capital T, which is actually tall. Capital T small t, which is actually again tall. Capital T small t, which is again tall. Small t, which is actually dwarf. So from this particular monohybrid cross, you will come across a ratio that is three plants or three progenies obtained are tall and one progeny is a dwarf. So 3 tall is to 1 dwarf. That is what we observe. Tallness or height of the plant is what we observe. So that is said to be the phenotypic ratio. And the phenotypic ratio of monohybrid cross, the phenotypic ratio of monohybrid cross is 3 is to 1. Okay, the phenotypic ratio is 3 is to 1. And at the same time, I'll draw you or help you with the genotypic ratio also. What is the genotypic ratio? What is actually meant by genotypic ratio? That is the genetic constitution. So one capital T is formed is to one two capital T is formed with one small t is formed. This is the genetic constitution. So the genotypic ratio is 1 is to 2 is to 1. Yeah, Calvin, it was given like 9. Oh, it was given like 9:30 p.m. Oh, really sorry, it was just a mistake. Okay, oh, I thought why you were not there, even though the class was about the genetic. I thought why you were a person who doesn't miss classes on genetics, and I have seen you. I thought you were late waking up. That's why I haven't spot about anything. Yeah, okay. I thought it was, yeah, it was in the morning. Yeah, the time when you saw that, you might have asked me, right? Evening, you have the test paper, right? So I got a few time in the morning, so I thought I should take the class in the morning itself. Anyway, about the monohybrid cross, right? We will be moving on back to the slides. Okay, don't worry. I hope you can understand this. Is it, Calvin? You know what is genotypic ratio? What is phenotypic ratio of the monohybrid cross? Is it? Okay. So back to the class. Inheritance of one gene. So what is monohybrid cross? We discussed in the last slide, that is monohybrid cross involves two parents or two plants differing in one particular character. So Mendel used one different character of two different contrasting traits. Okay. So tall and dwarf, yellow, round, axial, terminal, constricted, inflated. So one character was contrasted between two different plants. So I'll tell you with an example for the slide. I'll tell you homozygous tall we are taking as the parent, and homozygous dwarf we are taking it as the parent. Suppose this homozygous tall is the male and homozygous dwarf is the female. It can be alternate vice versa also. Okay. Calvin, if you have any doubt, just frankly open up and ask. I'll be free to explain that. Okay. Because it was a mistake from my side that I scheduled the timing wrong. Okay, so male can be dwarf also, female can be tall also, so vice versa. Since I have taught it like that, homozygous tall is male and homozygous dwarf is the female. So the gamete from this case is capital T is the gamete, and from this case small t is the gamete. So the F1 generation, you will obtain a progeny with capital T, small t. So heterozygous tall is the F1 generation or the first familial generation. And what we are going to do next one, that is F1 selfing. We are self-pollinating the F1 generation, that is capital T small t into capital T small t. The gametes are capital T small t, capital T small t, and we are getting a this one. Okay, so 3 is tall, this is tall, this is tall, this is tall, and this is actually dwarf. So in this case we are obtaining 3 tall is to 1 dwarf, which is a phenotypic ratio 1 capital T, 2 capital T small t, and 1 small t small t. So this is the genotypic ratio, this is the phenotypic ratio. Do I want to explain the cross once again with some other traits? Tell me yes or no on the chat box. If there is any issue with the audio, please tell me. Okay, I have reduced the volume of my device beside me, so I won't be possible to hear whether the audio is breaking or not. If it happens in between, but I've checked the connectivity, it's fine. So do I need to do any cross with any other traits? Please tell me on the chat box yes or no. Okay, now let's do an example. Now let's do an example with round and dwarf, round and ringled. Okay, so the parents taken as round, homozygous round into a homozygous round is crossed with the ringled. Okay, so what does actually round represent? Which letter is used to represent homozygous round? Calvin, on the chat box. Which is the letter which is used to represent homozygous round character? On the chat box, fast. Don't waste time. We can go in a very good way. We can enter and do the problems. You have concept videos at the end. What is actually the symbol for homozygous round? Homozygous means both letters should be same. Round means the dominant trait. Can you hear me? No idea. Guys, round is indicated with the letter capital R, right? So homozygous round means capital R, capital R. Ringled is the recessive trait, so small r, small r. These are the parents. Okay, these are the parents. These are the parents of in this case. So next you are going to take out the gametes. Okay, gametes from this case is capital R. From this case, small r is the gamete. So we are having a cross between this. You are getting capital R, small r, which is actually the F1 progeny or F1 generation. Calvin, is it clear? Next, you are going to do selfing. Next, you are going to do the selfing of F1 generation. Selfing of F1 generation can be understood clearly with the help of a Punnett square. That is, you can split out this capital R, capital R, small r. If you have doubt in this, you won't be able to understand any concept related to this chapter. So ask it right now if you have any doubt. So capital R, capital R, capital R, small r, small r. So what is actually capital R stands for? Calvin, I'm waiting for your reply. What is capital R? Capital R is what is the phenotype? Is it round or is it wrinkled or what is the phenotype? Capital R, capital R. Tell me fast on the chat box. Yeah, what is the phenotype? This capital R, this progeny would be what? Round or wrinkled? This progeny, I'm asking, this progeny, is it round or wrinkled? Absolutely, it has to be what? Round. This is to be round. This is also round because one dominant trait is present, so it is to be that trait, round. This is again round, and this is to be what? Wrinkled. Okay, round and wrinkled. So this is all about the monohybrid cross. Clear? So you have to have a clear idea about this. The dominant trait is to be expressed. Okay, dominant trait gets expressed. Phenotype is what we observe, that is tall, dwarf, round, wrinkled, white flower, violet flower. All these are what we see with our eyes. So they are the phenotypic ratio. Okay, what the observable character. Whereas the genotypic ratio is the genetic constitution, capital T, small t, capital A, small a, capital V, capital W, small w, capital W, all those things. Okay, so you have to by heart this particular point. You have to by heart this particular point. By heart this point, monohybrid phenotypic ratio. This is a previous year MCQ question, direct MCQ, but now we are not expecting this kind of a question because this was such a direct question few years back. Okay, 3:1. You have to study this also. This also 3 is tall and 1 is dark. You have to study it like that. Monohybrid phenotypic ratio is 3 tall is to 1 dwarf. Is it clear? So like that, monohybrid genotypic ratio is homozygous tall 1, heterozygous tall 2, and homozygous dwarf 1. So 1 is to 2 is to 1. Guys, this is what heterozygous tall. This is what homozygous tall, homozygous dwarf. Is that clear? Mendel made similar observation for other traits of character also. And right now we done the cross with what all things we done a cross with the height of the plant, that is tall versus dark we done, then white versus violet flower we done, violet versus white flowers we done, then round versus wrinkled seeds we have done. So Mendel actually made a cross with all the other things also, all the other traits: flower color, flower shape, seed shape, pot color, pot shape, flower position, and plant height. Flower color, seed color, all these character he used. He proposed, so after doing this experiment, he proposed that some factors, okay, some factors were inherited from parents to the offspring. So this particular factor, and this is the parent, this factor is passed on to the offspring. Factors are now known as the genes. Who call the term genes? Who call the term genes by Johansson? In the second lecture, we have studied factors were called by Mendel. So Mendel called the genes as factors, and the factors were called as genes by the scientist or the biologist known as Johansson. So the F1, when self-pollinated, produces gametes T and T in equal proportion. So F1 progeny produces capital T and small t in equal proportion. During fertilization, pollen grains of capital T have 50% chance to pollinate, and also the pollen grains of genotype small t also have a 50% chance. Guys, does each box contribute 25%? Okay, so what happens? There are two capital T, and there are two small t gamete form. So this gamete have 25 plus 25, 50%. So tall trait can goes into 50% of the progeny. Dwarf trait can goes into 50% of the progeny. That is what meant over here. Okay, you don't have to study all these things in detail because you have to have a clear idea on how to do the problem. Mendel self-pollinated the F2 generation. He found that the dwarf F2 plants continue to generate dwarf plants in F3 and F4 generation. This you will study in your degree level. Okay, in this class 12th or Neet base, you have to study F1 and F2 generation, which is important. Is that clear? Can we move into a video? Can we move into a video, everyone? Just give me hearts on the screen and let's move on to a concept video. Oh my god, the video has been stuck. Please wait for 1 second. Please wait for 1 second. Yes.
The Mendelian Cross is named for Gregor Mendel, a 19th century Augustinian monk who performed and documented the first controlled genetic process and allowed humanity to begin to understand the nature.
Listen to the video very carefully. They would be explaining you each and everything in detail.
A Mendelian Cross begins with a controlled breeding experiment spanning at least 3 generations and is designed to determine the inheritance pattern of a trait. Now here's some terminology to help us describe these crosses. We call the outward appearance of a trait its phenotype. The phenotype of this flower is purple. Each version of a gene is called an allele. The examples in this video will have 2 types of alleles. Dominant, represented by an.
So guys, listen carefully here. Listen carefully over here. The phenotype is the outward appearance. Okay, the phenotype is the outward appearance. Is the outward appearance. Alleles is one version of a gene, that is capital T, capital T. This one T, okay, it is the allele. Dominant allele is always indicated with an uppercase letter. Is always indicated with an uppercase letter. Let's continue.
Or recessive, represented by a lowercase.
So recessive is indicated with a lowercase letter. Look, dominant trait capital A, recessive trait small a. Genotype is a combination of genes.
The genotype of each trait is represented by 2 alleles. The next concept to understand is that every Mendelian cross begins with true breeding parents. True breeding means that if 2 individuals of that variety of organism are mated, then all the progeny will resemble the parents for a particular trait.
So guys, the parents we have selected, look, is the audio breaking? Is the audio breaking? The parent, both the female parent is also the.
Then all the progeny will flowered parents always produce. Start the Mendelian cross. Will be true breeding individuals.
Second, 1 second, clear into a hand. Second, please wait, everyone. Now is my audio clear? Now is my audio clear for everybody? Please tell me yes or no on the chat box. Yes. Now audio is clear. Once again, let me open the presentation. The system got hanged and everything itself by itself got closed. So let me open it first of all. In just 2 minutes, we'll resume the class. Okay, don't worry. Yes. Yes, can you all hear me properly? Just give me hearts on the screen, everyone, if it's clear. Yes, thank you so much. Yeah, let's back into the video. Oh my god, the system is getting hanged. Okay guys, so from that video onwards we'll discuss in the next class. Okay, because something is wrong. So whatever we have discussed today is all about the monohybrid cross. There was 2 to 3 problems also which I included. So that's all about today's class. See you in the next class with a wonderful session. Okay, in next class we would be discussing about the incomplete dominance in Snapdragon and also dihybrid cross starting we would be doing in the next class, and a few problems which is related to this chapter. Bye.